Hi, this is George with 573 Medicine, and in this video we're going to be discussing hematoxylin and eosin stain, as well as electron microscopy. So let's get started. So first, hematoxylin and eosin are stains which use two dyes in order to visualize histologic tissue. So hematoxylin is a basic or positively charged dye, and tissue which binds the hematoxylin will stain blue and will be described as basophilic. So it loves basic dye. The fill part is love, and then basophilic dye. So hematoxylin is positively charged, and so inside of a cell, the hematoxylin will bind to DNA because DNA has the negatively charged phosphate backbone. So DNA inside of the nucleus will end up staining blue. Eosin is an acidic or negatively charged dye, and materials which bind to eosin will be described as pink, or eosinophilic, because they love eosin dye. And the cytoplasm of cells will appear pink. So here we can appreciate the nuclear material, which is staining more blue, and we can also appreciate lots of cytoplasm, which is staining pink around the blue of the nucleus. So it's a better example here. We can appreciate the nucleus as well as surrounding cytoplasm. Next, let's talk about electron microscopy. And so electron microscopy is incredibly useful for examining incredibly small structures such as organelles, bacteria, and viruses. So going back, we can appreciate cytoplasm and nuclear material, but we can't really see things like the mitochondria or the cytoskeleton. And so that's where electron microscopy comes in handy. So there are two types, transmission and scanning electron microscopy. And first, we're going to be discussing how transmission electron microscopy works. So transmission electron microscopy uses electrons that come from the electron gun, and these electrons are shot and ultimately reach what is called a detector. So the fluorescent screening camera is basically the detector. And so we also have, between the electron gun and the detector, we have the specimen or the tissue that we're trying to examine. And so what ends up happening is that the tissue is composed of components that have different densities. And these densities are going to, these changes in densities are going to mean different amount of electrons ultimately reach the detector, and the detector is what gives us this picture. So the difference in densities creates these shadows that allows us to get pictures on transmission electron microscopy. And so transmission electron microscopy is going to produce a two-dimensional image, whereas scanning electron microscopy is going to produce a three-dimensional image. And the way scanning electron microscopy works is that it works in a similar way. However, the tissue samples are prepared in a way in which the detector can create or get electrons that produce a three-dimensional image. And so scanning electron microscopy, think 3D. Transmission electron microscopy, think 2D. And so that's it for this video. If you like the video, please like, comment, and subscribe, and feel free to email us anytime at realtalkmeded@gmail.com for comments, questions, or concerns.